You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Charlie Ren, head of the Ren Lab. Charlie, how you doing?
Hey, good, Richard. How are you?
Good. Yeah, tell me about your research. It looks, from the brief description, I'd rather you describe it than me, but it looks super interesting. Tell me what you're working on.
Sure. So I'm Charlie Ren from Biomedical Engineering here at Carnegie Mellon University. So our research is focused on, one of the major focus of our work is focused on lung engineering. So what we're trying to do is we're trying to engineer lungs using combining stem cells and biomaterials for transplantation therapy. One of the major needs we see in the clinics is, organ transplantation is almost the final option for end-stage organ failure. However, we simply don't have enough organs for the need. We don't have enough lungs. We don't have enough livers. So what we're trying to do is trying to develop a technology that we can engineer lungs with very similar structure and composition to native lungs for transplantation.
What makes the lung difficult to 3D print or to transplant or to work with? I would think, it's filled with lots of structures and alveoli, and it seems to be a very delicate organ. I don't know much about it, but, maybe give us a feel for what's unique and special about lungs and makes them challenging to deal with.
I think 3D printing, speak of that, is a very promising technology. It's a very different angle to approach a similar question. I think one thing we see, as you can see, the lung is primarily made of two parts. One is the airway part, where the air goes in and out, and the other part is the vascular part. So in the alveoli, which is the distal part of the lung, where there's a lot of little air sacs, is densely wrapped around by vascular cells. And if we go through a very conventional medical or biological processing called histology, you can cut a section of the lung. What you see is like a honeycomb structures. It looks like very random, a lot of air sacs. However, all those structures are actually very well organized because the air only goes through the trachea. Then the air will able to go through the trachea and then reach every single alveoli. And the blood, where in the body, the blood will exchange gas with the airway. All the blood is coming from the pulmonary artery. They will perfuse every single those little air sacs and then coming out of the pulmonary vein. So this is a very highly organized structure, which is so far still hard for us to recapitulate from, a synthetic approach. That's why our approach is trying to use something we call decellularized organ scaffolds to do this job. And we think because the dedicated structure and require very well organized structure to do the job as a lung to breathe, to exchange gas, we think, how about we start with a scaffold? Just like, we're trying to build a complicated house or building with very dedicated internal structure. Our strategy is to build the building by strip the dry wall out of the building, but keep the frame structures and then rebuild the dry wall, basically.
Is how much surface area does a lung have? And if you use a scaffolding to recreate, the interface where the blood and the oxygen get exchanged, would that take away from the available surface area? Or, does that mean that the scaffolding itself needs to act as a permeable membrane through which oxygen can diffuse?
Yeah, that's a great question. So I think a long time ago when I read a textbook, people say in the human lung, the gas exchanging surface equals to, I don't remember, maybe a football court or a tennis ball court. So it's a very large surface area. So with our technology, with the decellularization, we actually don't change that. So the only thing we do with decellularization is we remove the cells. So we get rid of the airway epithelial cells, are the cells covering the airway surface. We also get rid of the vascular cells. Then you may ask, why do you want to get rid of the cells and then later put the cells back? It's primarily from the immunological perspective. Eventually we'll probably, once we have a patient need a lung to be engineered for transplantation, we need to find another source of lung as a scaffolding. So the lung, for human use, very likely the lung either coming from another donor organ or coming from animal with a similar size match. For example, right now people are looking at pig lungs because the size and architecture of the pig lungs are actually very similar to human lungs. However, we cannot simply take a pig lung, give it to human because there's going to be a xeno barrier. There's going to be very strong immunological rejection. However, a lot of research in the past has shown that one of the main sources of the immune rejection is actually coming from the cells instead of the scaffold. So our strategy, by removing all the pig cells that keep in the scaffold, we dramatically reduce the immune rejection that can come out from a porcine material. Then we'll replace the porcine cells with human cells. And potentially, because of a lot of progress from human stem cell biology, we can actually potentially replace that with the patient's own cells.
Well, I was imagining the structure of the lung as you were talking, and I know it goes from, it's like a tree. It goes from a big trunk where the air comes in, and then it branches out and branches out and branches out. And the gas exchange occurs at the smallest level, right? The alveoli. Is that right?
Yes.
So when you're considering scaffolding and cells and, replacing parts of the lung, I guess the easiest part is the trunk part, the initial airway intake. And then as you have to have more finely detailed scaffolding, it'll get more difficult. And probably the most difficult part will be the actual gas exchange surface, the alveoli area, right, to recreate?
Yeah, so actually what we're trying to do is we're trying to do the recreation part to a certain extent, but actually a lot of the work has been done already by the scaffold. So once we remove the cells, actually the compartment, let's say there's an airway compartment, there's a vascular compartment, they're still intact. So after we remove all the cells, if we perfuse some cells from the trachea, which is the most proximal part of the airway, if we perfuse it into the lung, they can actually very easily reach the very distal part of the lung. Similarly, in the vasculature, if we perfuse vascular endothelial cells from the pulmonary artery or the pulmonary vein, they can actually reach the final capillary level. Of course, this requires a lot of optimization of the procedures, but that's actually pretty straightforward. We can do it.
Okay. So where is the current challenge right now? What goals are you working on, or what milestones are you working to hit? Tell me some specifics about them.
Yeah, so I can, we can break that to two parts. I can talk about a little bit about the current challenges about this whole, organ engineering or lung engineering based on the decellularized scaffold. So one thing is the cells we're using, because eventually the function of organ will be decided by the function of the cells. However, currently, even though there has been a lot of progress on the stem cell biology, still we're limited by our capability of getting a real good cells for lung reconstruction. So speaking of two sides, first on the airway side, there's a lot of progress on deriving different kind of airway epithelial cells or alveolar epithelial cells from IPS, but still many of the cells are limiting their function. There's still somewhat immature compared to our native counterparts. Similarly, on the vascular side, study from vascular biology shows that different organs actually have very different vascular phenotypes, and the lung has their own vascular phenotypes. However, right now, we're still using fairly generic endothelial cells for regenerating the vasculature, which has to be changed if we want to regenerate a functional lung with a good vascular function that can last for a long time. That's one limitation. The other limitation is...
Quick question about that. So you said you want to create IPS, so induced pluripotent cells or induced pluripotent stem cells. Are you saying that, I guess it makes sense, depending on the type of tissue you're using to try to induce the pluripotency in, if it's further down the hill, if it's more differentiated, it's harder to get the cells that you're looking for, right?
Yeah. So we have to, not only us, there's a big community working in this field, is trying to derive the right induction condition to educate the induced pluripotent stem cells to become the cells we want, ideally with higher efficiency. And then eventually we'll have to purify those cells. And then after purification, we'll have to develop the cocktail, growth factor cocktail, to maintain the cell phenotype and then deliver those cells into our scaffolding to make the functional tissue.
Huh. Interesting. I guess the source of cells you're using, they've been epigenetically changed, so maybe that's why some are easier to induce pluripotency and that's useful to you versus not, right? It's just a wild guess, but
yeah, actually there are some. I didn't see much on the lung, but I think that's definitely a possibility. People actually realize there's epigenetic imprinting in the cells even before and after they get reprogrammed to become iPS. Some iPS are easier to become certain cell type than the other. I think that's definitely one possibility. The other possibility is we simply need to do further more investigation to further develop protocols to further mature cells to be a equivalent cells compared to the cells we're finding in the native body. And then that's more. We know a lot about epithelial cells, and for the vascular cells, which are another big part of the lung, because the main function of the lung is exchange gas, to pass the gas from the airway into the blood, and then to let go the carbon dioxide. We really know very limited about the lung vasculature, about their molecular phenotypes, their cellular phenotypes, or their functional profiles. That really limit our capability of regenerating those functional lung-specific vascular cells for regenerating a good lung vascular function.
Okay. All right. Well, I interrupted you, but continuing on, what are some of the other challenges?
Yeah. So the other tragedy is, now I talk about, we need different cell types. We need them to be mature. Then the next question is, how do we bring different cell types together to do a single job, to make a good functional organ? If we say, what is the definition of the organ? It's a functional entity composed of all different kinds of cells, but they work in a very synergistical way. They're organized in a very good way so that they have a single or cohesive functional output. And then in the lung, we talk about lung epithelial cells, vascular cells. There are many other cells should be involved for engineer functional lung. For example, there are stromal cells like fibroblast cells, and many people believe that hematopoietic cells like macrophages may also play an active role during the lung regeneration. And then those factors haven't been put into the context very much yet. There has been some study, but not very comprehensive yet. So that's subject to some future investigations. And then once we bring in all the key players, now how do we develop a good culture condition that can keep all different kinds of cells happy? So in the biomedical research or in the cell manufacturing industry, people tend to usually we work with a single purified cells, and we know that we have to develop a specialized culture media to keep the certain cell to be happy. And then now we're going to be working with 5, 10, or even more cell types together in a single organ. How do we develop a good media to support all the different cell types? And how can we help the cells set up a communication with each other so that they can work together to have a good functional output? That's something, remains a challenge in the field. And then the next challenge is, eventually we want the organ to be transplanted. And then we need to develop a good in vitro evaluation system to tell us when the organ will be ready for transplantation. To do that, we probably need to develop an approach to functionally evaluate the organ from different aspects, and then eventually combine that with some pilot transplantation studies in animal models so that we can say when the organ will be ready for transplantation, and however, on some other occasions, when the organ requires some further maturation. I think those are the challenges I can see in the field.
Well, have you worked to create organoids? And I spoke to a number of researchers that they're creating like kidney organoids and heart organoids. They don't have all the normal functions of the full organ, but then they're able to test and see functionality and learn a lot more about how the organ functions without, overcoming the challenge of growing a whole one.
Yes, I think we actually have a little bit of work on organoid right now ongoing. I think organoid is a very good approach for regenerative medicine purpose. So one thing we can see is organoid is a very good approach to study stem cell function. As we just talked about, we eventually need cells to build the organ, and then we need functional cells. And how do we know whether the cell we're getting is really functional? Organoid is a good approach to evaluate stem cell function, especially on the epithelial cell side. A single epithelial cell can actually form a long organoid with branching architectures. So they can tell us whether the cell we're using is capable or not. And also right now there is a trend in the organoid approach is people are trying to make more sophisticated organoid. But currently, most of the organoids are still made of a single lineage of cells, either lung epithelial cells, or intestinal epithelial cells, or kidney epithelial cells. Right now, there are some efforts also as our interest is can we make a complex organoid composed of many different components. For example, can we make an organoid with epithelial cells, vascular cells, and stromal cells, and to really recapitulate a miniature lung culturing a dish, and use that as a testing platform to address many of the challenges we just mentioned earlier. For example, we can study, can we develop a better media to support all different lineages together? And can we develop a simulation approach to educate those cells start to communicate with each other? And also we can potentially answer the question, by having vascular cells and stromal cell support in there, can we actually stimulate a better regeneration of the lung epithelial cells? I think organoid is a very good approach to help us to address many of those challenges.
You mentioned a couple of times that there's a lack of understanding of what is it, the microstructure of the lung, or the vascularization, or the physical details of the lung itself, or where is the lack of understanding right now of the organ?
So one big lack of understanding, I think, lies in the lung vasculature. It's partly related to my background because during my Ph.D. I was doing vascular development using a fish as a model, actually. And then, during my postdoc, I was working on vascular regeneration. And then when I tried to combine all the previous training I had, all the background I have, I realized one of the major challenges in organ engineering is to engineer vasculature, because without a vasculature, we will not be able to transplant the organ. And many organs, if we look at the organ's function, is actually trying to deal with blood. So let's think about it, in the lung, we're trying to exchange gas with the blood, right? And then in the liver, we're trying to perform metabolic functions to process metabolites in the blood. And then if we think about the kidney, is actually trying to do filtration function with the blood. Actually, many organs, their key function is to interact or process blood. So that's why blood vessel is such a key component in our body. However, there has been a lot of knowledge, traditional knowledge from 20, 30 years ago or even earlier. We know that different organs have different vasculature. I can give you 3 examples: the lung, the intestine, and the liver. The lung's capillary vasculature is very tight. We call it a continuous, non-fenestrated. That means there's no gap between vascular cells, and then inside the vascular cells, there's no tiny windows. So generally, the lung vasculature is very tight. Otherwise, we're going to have fluid enter the airway. We're going to get drunk. We cannot breathe anymore. So and then the other example is the intestine. Intestine, their vasculature is, we call it continuous fenestrated. The intestinal vasculature, they have no gaps between cells, but they have windows inside the cells. And then go further down, the liver vasculature, we call it discontinuous fenestrated. There are big gaps between cells. There are windows inside the cells. It's very interesting to us from many angles, especially from developmental biology and stem cell biology angle. The reason I say that is because the lung, intestine, and liver, they all come from the endoderm. As you know, we have 3 germ layers: endoderm, ectoderm, mesoderm. All the 3 organs are from endoderm. However, they have 3 distinct microvascular phenotypes. I think from organ engineer perspective, we have to recapitulate those organ-specific vascular phenotypes in order to engineer a good functional organ. However, right now we know very, very little about this.
Wow. The more I learn about the human body, and I'm sure you experience this, it's unbelievable how complex it is and how it all functions. It's just amazing.
Right. But I think we're definitely, people are making progress in the field. We saw one problem that actually helped us uncover a deeper challenge for us to conquer, and then we can gradually bring the organ function closer comparing to their native counterparts.
Well, just a couple more questions on this. What are some surprises that you've gotten, over the last year or two in your work, maybe that inspired a new direction, or you made you think, oh man, this is even harder than I thought? Like what surprises have come about recently?
Let me think about it. So one surprise, when we work on this organ engineering based on the organ scaffold is the cells actually, once we use the right cells, once we give the right condition, the cell actually have amazing self-assembly capability. They can actually build the structure we wish them to build as long as we give them the right condition. I think we just talked about an organoid, which is a great example. You can just put a single cell into a 3D hydrogel. They will just make that beautiful structure, even differentiate inside that structure. I think that's one of the surprises I encounter when we work on this. So we see this many times in epithelial cells and also in vascular cells. And then what that leads us to do is, how can we provide the right environment to maximize the self-assembly capability of the cells? So I want to make that same analogy we just mentioned earlier about how to build a house. So our approach, we're trying to build a house based on the framework of the house. But however, usually when we build a house, we're going to have constructors going to the house to put on the wall, put on the appliance, all kind of things in there. However, when we engineer organs, we cannot send anybody into the organ to physically put this cell here, that cell there. We have to rely on the cell itself to find a way to go there and to build the structure they want to build. Actually, like I said, the surprise, the cell can actually do this. It's amazing. We work a lot on the vascular engineering in the lung. So if you flow vascular endothelial cells as individual cells, like a ball, every cell is like a sphere, flow into there. Once we give the right stimulation growth factors, medias, the cells can actually undergo self-organization, remodeling. Eventually, a vascular tunnel with a lumen inside, perfusible tunnel will show up in the vasculature. So that really highlights the capability of the cells we're using, and that also highlights the requirement of the efforts to give the right condition to induce the cell to do the right thing in the scaffold.
And I probably know the answer to this already, but when induced pluripotent cells are put into a matrix, if they're not directed to, it sounds like they'll tend to make the same structure over and over and over again, right? Like, or have you seen that they'll make different structures than what naturally occurs in the body once they've been brought back to the stem cell phase? Do they ever make different structures, or they always make the same exact structure that we see in the human body?
So let me try to get a better understanding of your question. Are you trying to say the stem that induced pluripotent stem cells usually tend to make the same structure where they are coming from? Like they may coming from a skin fibroblast reprogramming into iPS. Are you saying they tend to make skin fibroblasts, or you're saying if we put a stem cell into the lung scaffold, they tend to make a lung structure? If we put it into a different organ scaffold, they will tend to make a different organ structure.
Yeah, is the scaffold directional, or is it just like a scaffold and the cells literally know what to do? Like, for instance, the branching pattern of the of the lung. Do they always make that pattern, and does the scaffold need to inform them or guide them, or it just needs to be there and the cells just naturally know their own path and they make the right structure?
Yeah,
how smart does the scaffold have to be?
I don't think the scaffold itself is. I think the scaffold is more providing a permissive environment for the cell to adhere, to spread out, to build structures. I don't think there is much active induction role in the scaffold, even though I'm aware there are studies showing the scaffold provides some supporting or facilitating signals to help certain structure get built. That's possible. But I don't believe that if you put a cell, a more pluripotent cells, or the cell has multiple capabilities, if you put that cell onto a lung scaffold, it will become a lung cell, or if you put that cell into a liver scaffold, it will become a liver cell. I don't think that will happen very often. So that's why, when we put cells into a scaffold, we actually try to differentiate cells first. If we want to make a lung with a lung scaffold, we actually have to make lung cells first from iPS, and then put those differentiated lung cells into the scaffold, instead of relying on the scaffold to educate the cells to become a specific organ cells.
Okay. Yeah, I just wanted to get an idea for how smart the cells are, but they're
actually that's a question which we actually get asked a lot, from people when we discuss our research with, because it's such an intuitive idea. Earlier we have a lot of discussion when we enter the field and start to work on from the beginning. We're having a hypothesis: do we have a zip code in the scaffold? Does the cell recognize any special epitopes on the scaffold that will feed back on the cells to educate them to know what they should be? I think based on what I have seen so far, it's not that obvious. We have to kind of guide the cells through to educate them to become the cell type we want, and then put them into a scaffold. One key thing I think the scaffold is giving us is the anatomical architecture. example, like you said, the several branching airway structure, and also the branching vascular structure, and also the spatial organization between airway structure versus vascular structure. That's the key information we want to get from the scaffold.
Okay. Well, very good. So what's ahead for the next year or so, or a couple of years? What do you expect? What kind of milestones do you think you're going to hit, or it's just open-ended?
Yeah, I think from our end, we're interested in two major directions, which are related to this lung bioengineering. One side is on the stem cell, the other side is on the biomaterial side. So as you can see, when we make a lung out of the decellularized scaffold, we're actually combining cells with a scaffold. That's the key technology we're using. So on the stem cell side, I think we're hoping to come up with some idea that helps to address the vascular, organ-specific vascular issue. So we're trying to say, how can we make organ-specific vascular phenotypes by using stem cell engineering? By recapitulating the interaction between vascular component with the organ parenchyma during development from stem cell differentiation. That's one major direction we're working on right now. The other direction is about biomaterials, which are the decellularized organ scaffolds. I mean, we've been working with them for many years now. They're great at providing the anatomical architecture. However, we hope them to do more. We hope the scaffold itself can contain more instructive information that can really guide the stem cell to do what we want them to do. It's matching part of the question you just asked. Does the stem cell have a lot of intrinsic signals to tell the cell to become this but not that? Right now, no. But we hope they can do that to a certain extent later. How do we do that? We actually hope to convert the scaffold, the biomaterial, into a drug releasing platform. So we're developing some chemoselective modification strategies that can allow us to selectively conjugate growth factors or other instructional biomolecules, immobilize them onto the scaffold, so that once we deliver cells into the scaffold, they can interact with those signaling molecules, and then to guide the regeneration to happen more efficiently.
Okay. Well, very good. So what are some resources for listeners where they could find out more and, take a look at all the work your lab is doing and maybe get in touch?
We have a lab website. I'll be more than happy to share later. And also the lab where I came from, Dr. Harris' lab at Mass General Hospital, is really a pioneer in organ engineering. In his lab, not only people working on lung engineering, people also working on heart engineering, kidney engineering. They have also published in testing and composite tissue engineering. I think that's another good resource for getting some of the information.
Well, very good. Well, Charlie, thank you for coming on the podcast, and it's super interesting work that you're doing.
All right. Thank you so much, Richard.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.